Good morning, everyone. Today we see chapter number 8 Cell the Unit of Life. Need previous year questions. Now, the first question: which of the following cell organelles is responsible for extracting energy from carbohydrate to form ATP? This is the question, say the extracting the energy in the form of the ATP. So the powerhouse of the cell is considered as the mitochondria. We see the context from the NCRT is the determined as the mitochondria are the site of the aerobic respiration. They produce cell energy in form of ATP, hence they are called as a powerhouse of the cells. So this is defined in the NCRT. So we consider it as the extracting the energy from carbohydrate to form ATP is the organelle is the mitochondria. A question from NEET 2017. Now we see the second question. Select the mismatch. So what is the mismatch? First one is the gas vacuoles in green bacteria. Yes, it is present gas vacuole in the green bacteria. The large central vacuoles in the animal cell. So this is the mismatch. This is the wrong one. In the NCRT described as the plant and animal cell are different as a former possess the cell wall, plastid and the large central vacuole which are the absent in the animal cell. So what we see the in the animal cell which are the absents are the first one is the cell walls, second one is the plastids and the third one is the large central vacuole. So in these three are classified the plant cell, the animal cell, and in the animal cell the cell wall, plastid and the large central vacuoles are the absent one. So what is the answer of this question? The large central vacuoles are animal cell is the mismatch, and the protists are the eukaryotic and methanogens are the prokaryotic. This one is from the second chapter of this book. So we have to see the large central vacuoles in the animal cell is the mismatch. It in the NEET 2016. Now we see the third one. The select the wrong statement. The first statement is the bacterial cell wall is made up of a peptidoglycan. Yes, the bacterial cell wall is the made up of the peptidoglycans. Second one is the pili and fimbriae are the mainly involved in the motility of the bacterial cells. So this is the wrong one. Uh, the pili and fimbriae are not mainly involved in the motility of the bacteria. It described in the NCRT as besides of flagella, the pili and the fimbriae are also surface structure of the bacteria but do not play a major role in the motility. This is the IMP one. This is at the surface structure as long as the flagella, but the pili and the fimbriae are do not play a major role in the motility described in NCRT. So we consider it as the pili and the fimbriae are mainly involved in the motility of the bacteria. Or this is the wrong one statement. Answer is the B, and the cyanobacteria lack of the flagellated cells. This one is right one, and the mycoplasma is a wall-less microorganism. This is the most IMP and the remembering statement. It also come as the question. So we have to remember the mycoplasma is the wall-less microorganism. Now we see the fourth one. A cell organelle contain the hydrolytic enzyme is. So it is the lysozyme. This is the most simplest question from NEET 2016. In NCRT describe the lysozyme as these are the membrane bounded vesicular structure formed by a process of packaging in a Golgi apparatus. So the Golgi apparatus does the packaging of the lysozymes, and the isolated lysosomal vesicle have been found to be very rich in almost all type of the hydrolytic enzyme. So which are hydrolytic enzyme present? The hydrolase are the lipase, protease and the carbohydrate. So what is the lipase? Which done the digestion of the lipid called as a lipase. What is the protease? The enzyme which done the digestion of the protein called as a protease, and what is the carbohydrate which digests the carbohydrate called as the carbohydrates. Optimal pH at the acidic pH. So when the pH is the less than 7, it termed as the acidic pH, and in the acidic pH condition the hydrolytic enzymes are been activated, and it digests the cell structures and the blast the cell, destroy the cell. That's called as the lysozymes work or its conduct, and these enzymes are capable of the digesting carbohydrate because it had the carbohydrates, proteins for the protease, and the lipid digest by the lipase, and the nucleic acid because the nucleic acid have the also carbonic structure which digest by the hydrolytic enzymes. So a cell organelle contain the hydrolytic enzymes are the lysosomes. Now we see the fifth one. The mitochondria and the chloroplast are first statement is the semi-autonomous organelles. And the second one is the formed by the division of the pre-existing organelles, and they are contain DNA but lack protein synthesis mechanism. So these are the two statements. Now we see the first statement, a semi-autonomous organelles. This is the right one. Why it is the right one? Because mitochondria and the chloroplast has its own DNA. The DNA form the RNA, and the RNA and the ribosome also 70S ribosome. This is the most IMP. The 70S ribosomes present in these cells. In this organelle, in this organelles, mitochondria and the chloroplast have the 70S ribozymes, and the DNA, its own DNA present in these organelles. So they are from their protein and the other mechanism done by this cell, but some mechanisms and the some processes of these organelles also dependent on the cell nucleus. So the mitochondria and the chloroplast are the semi-autonomous organelles because it also depend on the cell nucleus. But second statement is formed by division of the pre-existing organelles. These are the right one. The mitochondria and the chloroplast both are formed by the pre-existing organelles, and they contain DNA. Also they contain DNA. This is the right one, but lack of protein synthesis mechanism. This is the wrong statement. The lack of protein synthesis mechanism, this is the wrong one. Why? Because the mitochondria and the chloroplast have their own DNA. So the form of the DNA, the DNA produce mRNA, tRNA, and rRNA, and also have the ribozymes. Which one ribosome? The 70S ribozymes also present in there. So they synthesis some proteins, and these proteins are helped to pro- process all the mechanism of the mitochondria and the chloroplast. So the statement is the wrong one. The lack of protein synthesis mechanism. So which one is the right one? The answer is A. The A is true. These are the semi-autonomous organelles. This one is the true statement, and the second one is the formed division of the pre-existing organelle, and they contain DNA. From on this side, they are this statement is right, but the wrong one is from lack of protein synthesis. So B1 is false. So the statement A is the answer. Now we see the sixth one. Microtubules are the constituent of the which one? The first one is the centriole, spindle fiber, and the chromatin. So it is not described properly in the NCERT book. So the microtubules are constituent. Constituent means it is a base structure of which organelles of the cell. So we have to remember that the microtubules are a constituent of the spindle fibers, the centrioles, the basal bodies of the cilia and the flagella, the chromosome fibers and the sperm tails. These are the five constituents where the microtubules present, and the other ones are also present. So now we see the first one is the centrioles. Yes, centrioles are present. The microtubules, spindle fibers. Yes, the spindle fibers also constitute the microtubules and the chromatin. Now the chromatin is the wrong one. Chromatin doesn't consist the microtubules. Now we have to see the second one, the centrosomes. The centrosomes have doesn't have the microtubules. Nucleosomes doesn't have the microtubules, but the centrioles all have the microtubules. Now the cilia and flagella. Yes, cilia and flagella both have the microtubules, but the peroxisome doesn't have the microtubules. Now we see the spindle fibers. Yes, spindle fibers have the microtubules, centrioles have the microtubules, cilia has the microtubules. So this D is the right one. This is the question from NEET 2016. So the microtubules are the constituent. It is a present in spindle fiber, centrioles and the cilia. Now we see the seventh one. Which one of the following cell organelles enclosed by a single membrane? So now from this question we have to see the yes this information. First one is the non-membranous organelles. Second one is the single membranous or bounded organelles, and the third one is the double membrane bounded organelles. So we see the all three one. First one is the naked or the non-membrane bounded organelles. So these organelles do not have any membrane. So this structure doesn't consist any membrane around them. So first one is the ribosome, second one is the centrosome or the centrioles, third one is the microtubules means the microfilament intermediate filament called as the cytoskeleton. The fourth one is the nucleus and the fifth one is the chromatin. So all five, ribosomes, centrosomes or the centrioles, microtubules or a microfilament intermediate filament called as a cytoskeleton, the nucleus and the chromatin, these five doesn't consist any membrane around them, so they are called as the naked or the non-membranous organelles. The second one we see the are single membrane bounded organelles. So which one are the single membrane bounded organelles? These are surrounded by the one unit membrane, a single unit membrane around them. The first one is the endoplasmic reticulum, ER. The both one are the rough endoplasmic reticulum and the smooth endoplasmic reticulum, both consist the single membrane around them. Second one is the Golgi apparatus. Golgi apparatus also consists the single membrane around them. Third one is the lysosomes, one layer. Fourth one is the peroxisome, and the fifth one is the vacuoles or the vesicle or the endosomes. Also, these are the single membranous structure. So we have to remember that the lysosomes in the single membranous structure it formed from Golgi apparatus, but the ribosomes which are also formed from the Golgi apparatus or it bounded by the Golgi doesn't bounded by the Golgi apparatus. So these are the non-membranous structure called as the ribosomes. Now we see the third one, the double membrane bounded organelles. These are the two membranes are the outer and the inner one, often related to energy transformation or the genetic material. The genetic material are the nucleus, mitochondria also consist, and the plastids. Which plastids are the chloroplast, chromoplast, leucoplast? These plastids, amyloplast also consist are the double membranous structure, nucleus, mitochondria and the plastids. These three are considered as the double bounded structure. So now what is the question? Question is the yes this one. Seventh one, which one of the following cell organelles enclosed with the single membrane? So now we see the single membranous structure is lysosomes, mitochondria the double layer, nuclei is the double layer, chloroplast also double layer structure. So we have to add in this one double membranous boundary structure is the chloroplast is there. Okay. Now the ninth one. Which of the following structure is not found in a prokaryotic cells? So prokaryotic cells is which are the prokaryotes? So the in NCERT described as the cell that have the membrane bounded nuclei are called as a eukaryotic, where the cell that lack of membrane bounded nuclei are called as a prokaryotic. An overview of the cell described in the NCERT have this statement in it that the prokaryotic cell is a lack of membrane bounded nucleus, where the eukaryotic cell is considered as the which cell has the membrane bounded nucleus, double layer membrane bounded nucleus. So the nuclear envelope is doesn't present in a prokaryotic cells. So not found in a prokaryotic cell are the nuclear envelope, the mesosomes also present, the plasma membrane also present, and the ribosome. Which ribosome? The 70s ribosomes present in the prokaryotes. Now we have to see the 10th one are the cellular organelle which cellular organelle with the membranes are so with the membranes are the endoplasmic reticulum, ribosomes and the nuclei. So the ribosomes is the naked one. So the first one is the wrong statement. Second one is the lysosome, Golgi apparatus and the mitochondria. So this is the right one. The lysosomes also have one layer. It considered as the membrane, not considered as the single one or the double one. So the lysosomes are the single layer, Golgi apparatus is the single layer, and the mitochondria is the double layer structure. We see earlier it like this one, the lysosomes and the Golgi apparatus are the single layer structure, where the mitochondria is the double layer structure. So the second one is the right one. These structure are the membranous structure. The third one is the nuclei, ribosomes and the mitochondria. So the ribosome doesn't is have the membrane, so it is the wrong one. And the chromosome is present in the nucleus, it doesn't bounded from the any membrane. The chromosome doesn't have its own membrane, the nucleus have the membrane. So the chromosome is also a wrong statement, it doesn't have the membrane, it's the naked one. And the ribosomes also doesn't have the membrane. So these two are the wrong ones. So the right statement is the lysosomes, Golgi apparatus and the mitochondria. Now we have to see the 11th. The which of the following are not membrane bounded? Also a simple one. Ribosomes are the not membrane bounded structure. The lysosomes is the single layer. Mesosomes is the single layer. Vacuoles is the single layer. Now we have to see the DNA is not present in ribosome. The simplest one. The nucleus have one DNA present, the cell DNA present in the nucleus. The chloroplast have the own DNA, mitochondria have its own DNA. So it 2015 NEET question. Now we have to see the 13th one. The nuclear envelope is derived of microtubules, rough endoplasmic reticulum, smooth endoplasmic reticulum, and the fourth one is the membrane of the Golgi complex. So we have to remember the diagram of the NCERT. The NCERT diagram shows the nuclear envelope has the derived from the rough endoplasmic reticulum. This is the structure. This is the nuclear envelope and the rough endoplasmic reticulum is there. So these are the derivatives of the rough endoplasmic reticulum. So this is the most IMP. The nuclear envelope is derivatives of the rough endoplasmic reticulum. Now we have to see the 14th. The structure that are formed by a staking of a organized flattened membranous sac in the chloroplast. So the what are the flattened membranous sac organized the staking of the flattened membranous sac The staking of the flattened membranous sac. So we have to see the diagram. The staking of the flattened membranous sac are called as the granum. Okay, so the answer one is the grana. So what is the stroma lamellae? We have to see the whole structure. The first one is the flatten. The flattened structure is called as the thylakoid. This one, the thylakoid, is the first structure, the basic unit of the chloroplast. And the now the thylakoid are the staking or organized flattened membranous sac, and now they are the form granum, and each granum is connected with the one another is called as the stroma lamellae, as seen in this one. What it's called, stroma lamellae, which connected the granum or the single unit called as the thylakoid. And the stroma, which one is the stroma? The space in the chloroplast is called as the stroma. Okay, so we have to remember the structures name. Our first one is the thylakoid, a single flattened unit, and they together make it called as the granum, and the whole structure called as the grana, and the grana connected with the stroma lamellae, and the space in the chloroplast is called as the stroma, or the surrounding any space in the structure or the of organelle called as the stroma. It also present in the mitochondria, also present in the chloroplast. The space in the organelles called as the stroma. Now the 15th one, select the correct matching in the following pair. The rough endoplasmic synthesis of the glycogen. Sorry, this is the wrong one. Rough endoplasmic reticulum oxidation of the fatty acid. The smooth endoplasmic reticulum one. The smooth endoplasmic reticulum oxidation of the phospholipid. Sorry, this is the wrong one. Smooth endoplasmic reticulum synthesis of the lipid. This is the right statement, also described in the NCERT as where it's gone. No, it doesn't consist, but we have to see the, yes, these are the present. The rough endoplasmic reticulum frequently observed in a cell activity. It involved in a protein synthesis and the secretion. The rough endoplasmic reticulum involved in the protein synthesis and its secretion. They are extensive and the continuous with the outer membrane of nucleus, and the smooth endoplasmic reticulum is the major site of the synthesis of lipid. In the animal cell, lipid like the steroidal hormone and the synthesis of synthesized in the smooth endoplasmic reticulum. So this is the most IMP one, synthesis of the lipid done by the smooth endoplasmic reticulum, and the synthesis of the protein and the secretion of it done by the rough endoplasmic reticulum. So what statement is right? Smooth endoplasmic reticulum, synthesis of the lipid. So this is the question from NEET 2015. Now we have to see the Golgi complex play a major role in the Golgi complex play a major role in as the energy transforming organelles. So this is the wrong one. It doesn't play as the role of the energy transforming organelles. Second one is in the post translation modification of a protein and the glycosylation of the lipid. So this is the, so the protein is developed by ribosome and the mRNA. This is the ribosome, ATS ribosome. Okay, and the protein is developed. So the process of the developing protein from mRNA and the ribosomes called as the translation. And after this translation, after the translation called as the post translation. So the post translation question is the post translation modification. So the PTM, the post translation modification of the proteins and the glycosylation of the lipids are done by which organelles? Golgi complex. Okay, so the right answer one is the B. The Golgi complex play a major role in the post-translation modification and the glycosylation of the lipid in the trapping of the light and the transforming into a chemical energy. This done by the chloroplast in the digestion of the protein and the carbohydrate done by the lysosomes. So the right one is the second one. Now we have to see which of one of the following organelles in the figure correctly match with the function. So the figure consists the that one part of the nucleus and this one part of the rough endoplasmic reticulum. So the nucleus is there? No. The Golgi apparatus is there and the rough endoplasmic reticulum is there. So now what is the function of the rough endoplasmic reticulum? Done is the protein synthesis. So this is the simple one. The what is the answer? The rough endoplasmic reticulum and the protein synthesis is the answer. Simplest one, need 2013 have this question. And the rough endoplasmic reticulum from the glycoprotein? No, the glycoprotein is formed by Golgi apparatus. Okay. Now we see the 18th one. The term glycocalyx is used for. So glycocalyx, the glycocalyx is the outer layer, most outer layer of the bacterial cell wall. The first one layer is the glycocalyx, the second layer is the cell wall, and the third layer of the bacterial cell is called as the membrane, cell membrane. So this is the cell wall, this is the cell membrane, and this is called as the glycocalyx. A layer present between the cell wall and the membrane of bacteria. So this is the wrong one. The cell wall of the bacteria? No, the cell wall of the bacteria is not considered as the glycocalyx. The third one is the bacterial cell glyco engineered to possess and glycosylate protein. No, this is the wrong one. A layer surrounding the cell wall is of the bacteria. So this is the answer. The most outer layer is the glycocalyx, so it surrounds the cell wall and the cell membrane too. So it a question from the Karnataka NEET 2013. Now we see the 19th one. Which of the following type of the plastic doesn't contain stored food material? So in this, for which doesn't have the food store in it. So now we see the plastic. So which how many plastid types are there? So four types of the plastid described in NCERT. The first one is the chromoplast. The chromoplast doesn't store the food. It have the chlorophylls and the carotenoid pigments which trapping the light energy from the for the photosynthesis. So our answer is the chromoplast, but we see also see three ones are the elioplast. So what is the elioplast? Is the elioplast store the oils and the fats. The aleuroplast store the protein, and the amyloplast store the carbohydrate. So the answer is the chromoplast, but the elioplast does the oil and fat. Aleuroplast consists the protein, and the amyloplast consists the carbohydrate as the form of starch. Now, what is a major site of the synthesis of the lipid? So, the synthesis of the lipid done by the smooth endoplasmic reticulum, NEET 2013. Now, the 21st are the select the correct statement from the following regarding cell membrane. So these all four statements regarding to the cell membrane. The first one, Sodium and the potassium ions move across the cell membrane by the passive transports. This is the wrong one. It done by the active transport. The protein make up of the 60 to 70% of the cell membrane. No, the protein have the only a 50 to 52% in the cell membrane. The other 40% is the lipid and the remaining is the other structure. So the protein is the only a 60 to 70%. This is the wrong statement. The third one is the lipid are arranged in the bilayer with the polar head toward the inner side. No, this is the wrong one. The polar head is present the outer side and the tail is present the inner side. So the inner part have the tail one and the polar part has the outer side. So this is also a wrong one. And the fourth one is only right. A fluid mosaic model of the cell membrane was proposed by the Singer and the Nicholson. So this is the right statement. The Singer and the Nicholson proposed the fluid mosaic model of the cell membrane. Okay. Okay, done. Thank you.